Hello, and welcome to day 2 of the Mitosis Notes. There'll be a total of 4 days of notes plus some worksheets. So, we kind of talked about the basics of chromosomes and DNA and prokaryotic cell division, but now let's step it up another notch. So here, this is going to be mitosis of a eukaryotic cell. And it's eukaryotic, you can tell, for a couple of reasons. One, you can see these things called centrosomes or centrioles. They're right there. And you can also see there is a nucleus. Prokaryotes do not have a nucleus. So you're still in interphase here, and you know you're in interphase because you still have this kind of spaghetti of DNA called chromatin. So it's not quite ready yet. It hasn't condensed. So you know you're not in mitosis yet. So here you've officially started mitosis right there. So in prophase, and again, we're going to talk about these coming up, but prophase, you see that there's some condensation of the DNA right there called the chromosome. You can see these things are starting to form. They're called spindle fibers. We'll talk more about that in the next video. And as you see this progression, you end up with what is soon to be 2 cells. We're, again, going to talk way more in detail about that in the next video, but I just wanted you to kind of see what you're up against for today. So DNA and cell division. So before we do anything, and we talked about this in the DNA topic, in order to make one cell into 2 cells, you first have to copy the DNA. So that was DNA replication, which you know that. You cannot simply split it. It doesn't work that way. And especially in your cells, this DNA is going to be broken up into these things called chromosomes. Now, if you are replicating a skin cell, you got 46 of those. Now, if you looked in that last picture, the chromatin is a disaster. It looks like a bowl of spaghetti. It's no good. So in order to organize the DNA, you need to condense it. And you need to condense it around these proteins called histones. And I'll show you a picture in a little bit. And then the whole package deal of the DNA and the histones is referred to as a nucleosome. So together, it's nice and neat. So let's take a look. This is a replicated chromosome. And if you remember from the DNA unit, these are going to be called sisters. And you know they're sisters because they're identical. And they use the word chromatid here and the word chromatid here, but of course you could put sister on top of that because they are sisters. They're held together at this thing called the centromere, which we talked about. But now if we zoom in, you can see the DNA is this double green line there, but it's only really one, but it's just double wrapped. The histones are these blue kind of, it's a real like an 8-pack of what look like balls, but they're not really balls. But so the histone is this protein, and it's positive because the DNA is negative. So the DNA, which is negatively charged, is drawn to the positively charged histones. They wrap around. And that nucleosome, which was the last word on the last slide, is this package deal of the DNA and the histones it wraps. So this is a very detailed picture, but when I draw a chromosome, I draw it like that. So if you see my picture like that, it's supposed to be that. Again, it's just a simple way to do it. You're not going to need to get involved with any of these details, but I just wanted you to see that DNA is extremely small. I don't know if you know how small a nanometer is, but it's 10 to the minus 9 meters. So again, I don't know if you can appreciate how small that is. So as the DNA starts to kind of organize itself or condense, you end up with a chromosome. Now, a chromosome is 1,400 nanometers. You can actually see this under a microscope. DNA, no way, too small. Again, you're not going to need to get hung up with these words and these numbers, but I just want you to see in case you care about the size. So here it is again, and this is what we talked about already. Whoops. We know that your sister chromatids are right there. We know that they're exact copies of each other, and we know that they're held together at a centromere. Okay, so we know that. And as I've said many times, it must be replicated or copied first. If it's not replicated, it cannot divide. It's not going to work. We can, but you won't have enough chromosomes, and that'll be a problem. Chromosome structure, as I said, is normally this kind of bowl of spaghetti mess that's called chromatin, and it was that kind of purple bowl of spaghetti in the first picture. And what's going to happen is it's going to start to condense, and again, I draw it like this, and that's including the histones in the DNA. And when it's replicated, you get sister chromatids, and they're held together at a centromere. So all that stuff is review. And the new word there is these special proteins are called cohesins, kind of like a cohesin. They're going to hold the two sisters together. So that's sister number 1, and that's sister number 2. They're held together with these cohesins at the centromere. So that's important. Now they're going to break up in the video tomorrow, but right now they're still linked. So in this case, this is a little bit more of a detailed picture. So we're going to see more about this coming up later, but these are known as homologous chromosomes. Now what it means to be homologous is they are the same chromosome number. So let's say that this is dad's number 3. This is mom's number 3. So that's what it means to be homologous. If you look here, when they're linked, see the two blue ones right there? Those two are sisters, and these two red ones are sisters. But since you have red over here and you have blue over here, they are considered homologous. So they have the same genes in the same spot. They're just not identical. There's our cohesin proteins, which hold together the sisters, which we talked about. And then there's these really cool things called kinetochores, and what they're going to be responsible. They have motor proteins, and these motor proteins, which we'll talk more about when we do anaphase, are what allows the chromosomes eventually to separate from each other. But we'll talk about that in the next video. Now the cell cycle, which we've talked about this already, the G1 is when the cell is going to grow. The S phase is when you're going to replicate your DNA, which we talked about. G2 is when we're going to prepare to divide, and what I mean by that is make sure that all the replication is good and everything's fine. And if you remember, those three together, the big piece of the puzzle, is called interphase, and that's the longest in the cell cycle. And then you have that little piece of the pie called mitosis, which again we'll talk more about in tomorrow's video, but that's where you're going to actually divide your cell. Now some cells, they don't go through the cell cycle. Things like nerve cells. Once you're born, the nerve cells you have, that's all you got. So that's why you can't really damage your brain by doing drugs and really traumatic brain injury, because if you damage those cells or a stroke, they don't come back. They are known as G0. It's not go, it's G0. I may also write it like that, as G0. So a cell that does not divide ever is in what is known as G0. So when that one dies, it dies. So be careful. That's why take your brain seriously. So G0 cells do not divide. If it's going to break off into G0, once again, it'll go right there out of the G1 stage, if it's going to. But here's again the cell cycle. And again, we'll just review. Grow and DNA replication. And then we're going to prepare here. Once again, that whole kind of beigey, pinkish color is your interphase. And then finally, the blue is your mitosis or your M phase. Now, the cell cycle, the length depends on a lot of things. So like the cells in your mouth, and let's say the cells of your skin, and you'll notice if you've ever cut the inside of your mouth or you cut your skin, the cells there have a very short cell cycle. They're going to replicate pretty quick, usually within a couple of days. Think about any time you've got cut. Certain things like your red blood cells, although they don't go through true mitosis, your body will make them at around 2 to 3 million a second. They're really small, by the way, so we make a lot of those. And just as we said, mature nerve cells, the ones that after you're born, they do not divide. They are in G0 until they die or you die. So cell cycle depends on the type of cell. Things like blood cells are made like crazy, and nerve cells do not divide. So if you're looking at the numbers, let's look at it here. So I'm going to make up a number, and let's say this is 1. So you got 1 chromosome. First thing you have to do is you have to take that 1 chromosome, and you're going to have to make it a double-stranded. Now, a lot of people think at this point you now have 2 chromosomes. You do not. You have 1 double-stranded chromosome, but you have 2 times the amount of DNA, and that is a very important distinction. You don't have 2 chromosomes, you have 1 that has been replicated. In this case, they're called sisters. Then, After cytoplasmic division, known as cytokinesis, you end up with 2 cells that each have 1 chromosome, and if all goes well, they will be exact. No mutations, hopefully. And then those can go on to grow and do it again. None of this can happen unless you have these things called spindle fibers or a spindle apparatus. And that is kind of these microtubules. And what happens is, and we'll talk more about it tomorrow, but at the end, I drew them sort of kind of like this. That's not what they look like. But there are these things called centrioles. And what's going to happen is a centriole, oops, my line crossed out there. Centriole is going to have these microtubules called spindle fibers, and they are going to be what is ultimately going to allow a cell to separate. But again, we'll see that more tomorrow. So the spindle fibers are these microtubules. They're going to extend from the poles. Once again, we'll talk more about that tomorrow. And at this point, they are going to allow the chromosomes to move with the help of these things called kinetochores. Now, both plants and animals will have spindle fibers. They just have a little different setup. In a plant, they originate from something called a centrosome, and in animals, they're called centrioles. Plants don't have true centrioles, but the idea is the same. And here's the last picture for today. This here would be your centriole right there. The pink represents the spindle fibers, and the purple, that actually represents the chromosomes. So here you can see this is going to be what's known as anaphase. And again, we didn't do this yet, but this is anaphase. And you know that because the purple, well, the red doesn't work out so good. The purple chromosomes are going that way and the purple chromosomes are going that way. So tomorrow we will pick up with mitosis and all the fun steps.